My name's Tullis Matson. I'm chairman of Nature Safe and founder. And I would just like to thank Victoria and the International Congress of Zookeepers for allowing us to give this presentation all about Nature Safe today. And it's about how we apply the application of technologies to preserve viable cells and gametes for animal conservation for future regeneration. So Nature Safe, our mission is, it says in the title, we are here to save animals from extinction by collecting and indefinitely storing reproductive cells and cell lines from endangered species. And this presentation is going to show you a little bit of the history, how we got there, and how we can sort of see the future is in living tissue bank preservation and the way that we go about this as well. As you're all acutely aware, we are facing the sort of sixth mass extinction at the moment, and it is of huge importance of finding different ways of stopping some of our rare and endangered species all around the world, finding new technologies to slow down, if halt or even reverse the extinction process. And a lot of this presentation is all about these sort of new technologies that are out there that we're using, not just to save rare and endangered species, but rare and endangered some of our native breeds as well. I'll show you that as well. So I've been working in the equine industry for 30 years and really been cryopreserving equine semen, and this is how it's really led on to this. And my passion myself is looking at rare endangered equine breeds and how we can stop them from going extinct by using these modern technologies. So, yeah, extinction is no longer a term just for wild exotic species. We look at our Suffolk punch that we have down here. There's less than 72 females left in the country, about 300 in the world. And these really do hold sort of certain challenges in just trying to save this breed from disappearing as well. And of 4 of the rare equine native breeds, 12 are considered rare, 5 are critical, with less than 300 females left. So we've really seen challenges, and we've used all different technologies to actually slow the extinction process of some of these breeds. Yeah, our rare and native breeds are seeing the same population challenges and threats to their survival as many of the zoo species as well. And I actually learned some of this terminology quite a few years ago at a conference at Chester Zoo about the extinction vortex. And we've seen this, how it's applied and how it comes into fruition really with various breeds. And we have the primary sort of threats at the time, which we see in the domestic side of our rare and native equines, of inappropriate breeding, inbreeding, which is really from the lack of awareness and understanding, and you know, some of the economic pressures. And that's seen our secondary threats. And here we see that when stallions come to us with issues with poor semen quality, smaller testicles, they have a low reproduction rate, they see a much increased post-neonatal death rate as well. And this, we can fairly put this down to some of these inbreeding issues that we're seeing. And we can see this obviously happening in another rare endangered species as well. And if we're not careful, we're obviously going to see on the equine world some of these breeds from disappearing. And it all comes down to inbreeding, and this is, you know, one of the big factors in making some of our rare breeds and species from disappearing. If we look at, you know, you look at something like the Clydesdale horse, they've got a 26% inbreeding coefficient. When you think of a brother-sister mating of 25%, you know, this is way above that. And we always have to look at inbreeding as not just what the inbreeding coefficient is, but the rate of that inbreeding going up. And I think it's really important that we have a look at that fact as well, because that can really spiral out of control. But there are obviously ways that we can turn this around and hopefully stop this these rare endangered breeds from disappearing by appropriate breeding. And now they have a traffic light system for the Suffolk horses, the Cleveland Bays as well. So they can really, like the horse Tinder of the dating world. So they try and mix breed matches. They use the Spark system, which I think originated from the zoo world many years ago. So they're very much on board with that, and they're seeing it start to turn around and having positive effects as well. By using a living gene bank, actually freezing down embryos and sperm cells, tissue freezing, and also we can export these genetics all around the world and help bolster up other breeds and stop that inbreeding from going out of control. So where does this really lead us? This really gave us the technology to say, I've been doing that for about 30 odd years, and then Nature's Safe was born last year, and Nature's Safe was a registered charity on about the 23rd of December last year. And what we were looking to do is to cryopreserve live the live reproductive and non-reproductive cells from endangered species. And this way that we can thaw these cells out and be used in assisted reproductive programs for the future. And so when we freeze these cells down, they're as good as the day that they froze them, as in 20, 30, 40, even 1,000 years time, they'll be still exactly the same quality. We've been very lucky. We've had some great press coverage since we launched back in December, but it was officially launched in the press in January. And this has really got us on the stepping stone of getting out there and making sure we get some of these tissue samples in, so we can start Nature's Safe off and start banking some of these species down. So, what is Nature's Safe? Nature's Safe is a collaborative charity, really bringing in all different parts of the zoo community together. We need your help basically to make Nature's Safe work and survive and thrive. And Nature's Safe, like you're saying, is a network of leading reproductive biologists, cryobiologists, and breeding management experts. You'll see in a minute all the people that are in the background and the infrastructure. And the science is moving on at such a rate now, it's quite difficult sometimes to keep up with that. So that's why we've got some great scientists on board trying to find out what it's going to be, not just for the next sort of year or so, but how we can cryopreserve these samples for many generations or years to come, and trying to second guess what those technologies are going to be. And we provide this service totally free of charge for zoos. So, without your help, we couldn't exist. So we need as many samples from zoos. As soon as an animal passes away, we can get those samples and sent to us so we can cryopreserve. And this is a free service at the end of the day for the zoos that are out there. So we're very much working with ER's a biobank, the ER's a management group, and we're a collaborative of expertise out there. So many other groups that we have. Chester Zoo has been quite fantastic, really. They've been right at the beginning and been in the background. And without them, it would have been very difficult to really propel Nature's Safe forward. As I say, we have now got an MOU signed with ER's a biobank. Other companies set up a charity. It takes a lot of infrastructure. It might seem the small things, but actually it's a very large thing. INT International, excuse me, they actually supply the labels to go on the cryovials. Now, when you've got not thousands but hundreds of thousands of vials, you want a good management system, and they're providing that with all the barcoding. So when these samples come in, we can know where they are, what the animals are, who owns those samples as well. So it's all from the human IVF side. So that's great to have them. CSAR, Stallion AI Services, our foundation company. Gemini Genetics, they have the technology on the tissue banking side. The Rhino Fertility Project will talk about this, and obviously EASA and Alpextra, they've very much been helping us and backing us in the background. And this is a short video of us just really endorsing Nature's Safe.
Hello everybody, my name is Richard Vine and I'm talking to you from the Old Pejeta Conservancy in Kenya. Behind us you can see the last two remaining northern white rhinos on the planet, and we are doing our best to recover this species from the brink of extinction. Both are infertile, and therefore artificial reproductive techniques are the only options that are open to us. Nature Safe is also playing a critical role in saving the world's biodiversity by creating one of the first living tissue banks on the planet. Their work is incredibly important, and we look forward to working with them in the future.
So that's a great endorsement from Richard there, really, about Nature Safe. So the aims of the charity, we're here to sort of develop technologies for cryopreservation. As I say, we're always trying to invest in these technologies to preserve these cells down, so when they're thawed out, they're in a useful state, so we can start growing cell lines, and doing cell culture from multiple different cell types, from multiple different taxa. So in other words, from certain mammals, it's fairly easy to freeze the cells down, but from certain amphibians, it's very, very difficult. So we need the expertise. We're to provide storage of living cells from endangered species, work along stakeholders, as we've said previously. It's very much, we want to involve as many zoos and many parties out there to really come on board. Without your help, we can't function. So we very much want you on board and helping with this. Obviously to enhance our network of expertise, to be able to sort of put this out there around the world, and employ all available resources and technology for cryopreservation, as we're saying, and to make sure that what we are doing can be used for the future animal conservation and the species survival. And when we got that knowledge, to disseminate that knowledge, this isn't just for us, this is to make sure that all people around the world can actually use this knowledge to freeze down cells and to be able to use those cells in a functional way. So who are Nature Safe? You can see some of these ones here I have to thank Rhianna Bolton. She actually put this whole presentation together and put all these slides together. It just shows you some of the animals that we've done on the way. You'll see some of them through the slides as we go. So yes, so the founder and chair of the trustees, but very much we have, as you see here, Dr. Susanna Walker, who is the head of science at Chester Zoo. She's been instrumental, really, of getting Nature Safe off the ground, and she's one of the co-founders as well and vice chair. Dr. Rhianna Bolton, who is also the co-founder and really was the person that helped get the charity off the ground right at the beginning and helped set up the charity the months beforehand, and actually works for Nature Safe now, either in the lab or as charity coordinator. And also Kate Ashmore, she's one of the co-founders and trustees and oversees the running of the charity. But it's not just down to 4 individuals. There's certainly a lot more players in this really to make it function. I say we've got Matt Pettit, he's one of the trustees. He's the one with IMT International who does all the cryovials and actually the data system behind it, which is so important. Professor Susanna Williams, gosh, she's the chief scientist, really looking into the new research and how we can make it work. And then we've got Val Edwards, who's one of the solicitors. We're obviously making sure that we're compliant with everything. And then we've got Nicole Gunter, who's obviously on the management side, Hayden, who looks onto the fundraising side, and Rob Humphreys, who's on the accounting side. And honorary secretary is Sue Jones. So those are really the trustees. But even behind that, we've really sort of split it into 2 halves, because it's got to be a fully functional charity, making sure we've got plenty of people on the fundraising, marketing, and making sure it functions well in an orderly manner. But obviously we've got to have the scientific and Zook stakeholders. So we've got an amazing team behind Nature Safe, really to propel it forward and hopefully to make sure it stays on the right tracks and reaches the goals that we want it to reach. So the science behind Nature Safe is so important. This is really what can make us different from a lot of other biobanks out there. So as I was saying before, I have about 30 years in cryopreservation on the domestic side, and that's, you'll see a few more slides on that, but that's really what's led us on to some of the technologies that we have here, everything from semen collection, semen storage, semen sexing, oocyte harvesting, and genetic preservation as well. So as I was saying before, many other biobanks, there's lots of other biobanks out there, but they quite often store at -80 freezers, where we store at -196. They store, just look at the DNA for conservation, genetic research, for future population management, whereas we're storing with cryoprotectants in there, so it permeates that cell membrane to preserve that cell membrane in a living state, so those cells can be stored indefinitely, and they can be reawakened, as we say, in many, many years to come. It always amazes me when you freeze, especially semen down, and it's so immotile, then literally 30 seconds of thawing it out, it's racing around the screen. It's amazing what we can do with cryopreservation and cryopreserves as well. And once these stores are stale, they obviously can be used in programs, breeding programs in the future, when we use the right cryoprotectants. So there's a few different ways that we can preserve these cells. So we can do something called post-castration epididymal sperm extraction, quite a mouthful, that one, testicular tissue harvesting, ovarian tissue preservation, and skin sample preservation. So we take the cells from different sites. The ear is one of the most popular sites because this is where we can get the cells grow very, very well from the ear from mammals. And the beauty about that, we've got up to 5 days of this tissue sample as well, after the animals passed away, for when it comes to the tissue samples. For the gonads or the reproductive cells, the ovaries and testicles, they've got to be a bit quicker. So really 24 hours, we want to get the tissues from those, or the eggs from those, the ovaries. But if it's sperm we're needing, really it's got to be within under 24 hours. So they've got to be a little bit quicker when we're doing the epididymal side. So these are the different sites that we can use from the different areas of the animal. So just with the epididymal, I say it's fairly straightforward. It's supposed to be doing a bit of time, but it takes it's not, it depends on the animal itself. If the bigger the testicles, the easier it is to do, basically. But we extract the semen from the epididymis, and yeah, we spin it down, and then if need be, and then we resuspend it in a freezing with cryoprotectants. We tend to use something called DMF, dimethylformaldehyde, which permeates the cell membrane a lot quicker than the conventional glycerol. And yeah, we freeze it down and thaw it out, and then the sperm cell should come back to life, and then we can use this in assisted reproductive techniques, like for insemination, for instance, or ICSI in the future. So we've done quite a few of these now. We've done about 150 different, I think this is even maybe up to maybe 200 different extractions now on the equine side, and about 45 of the non-domestic, non-equine species as well. And so here, we literally, this is the epididymis here that we take off, and then we can extract the semen from that. They tend to arrive chilled. We don't want them too cold, but we certainly don't want them warm either. And we get them ideally under 24 hours to get the best results. We have done some a little bit older than that, but generally, the quicker they get to us, the better. We literally wash the testicles off, remove the epididymis. And this process, it depends on the species itself, but this can take anything from 2 to 4 hours to do, because we dissect the epididymis on here, and it starts off about 3 or 4 inches long, ends off about 6 or 7 inches long by the time we dissected all the blood vessels off. And then we flush out the epididymis with an extender, and if they're big enough to do, and again, the bigger the testicles, it's easier to do. So we flush out all these vessels here, and the semen can come out. And then some species we can get between 10 and 60 insemination doses from a pair of testicles. But again, it does vary from species to species. So the species that we've done so far, we've done various species from outside the equine world, as you can see, from mouse deers to Kirkdicks to to leopards and cheetahs. So they all have their own challenges, I have to admit. This is one that we did quite often pre-freezing. You can see the sperm quite often are immotile, but we have to add something to it to actually wake them up. We add something called SSPR. We call it a synthetic seminal plasma replacement. And this actually wakes the sperm cells up post-thaw, which is great. So it's exactly the same sample. As you can see, post-thaw, it looks really good. And actually, when we did quite a few equines, and then we got this bovine in, we've never done bovines before, and actually these testicles were nearly 48 hours old by the time they came to us. And the semen quality wasn't the best. And we thought, well, we'll give it a go. And we did, and lo and behold, we had some calves on the ground from this method. So that's the light bulb moment. We thought, well, if we can do this on canines and not canines, bovines and equines, why can't we do this on other species? And that's where we started now to transfer the skill over from the domestic side onto other species. And from that one pair of testicles, they've got over 30 offspring now from that. That isn't outside the bracket. We wouldn't normally expect these good results from testicles at 48 hours old. So this is not the norm. We normally like them under 24 hours old. So this is an onager that we did quite a while ago. We got actually 29 past doses, and the post-thaw in it, as you can see here, is quite brilliant. The post-thaw was absolutely fantastic. Obviously, it's an equitor. We were fairly easy for us to handle and do, but it worked very, very well indeed. We did a cheetah. I think this is back in 2019, I think. Yeah, we managed to get some sperm cells from it. And this was post-thaw again. There wasn't many there, but what was there was moving fairly well. We were fairly impressed actually with the post-thaw on this cheetah that we did. We only got 6 straws from it, but at the end of the day, quite often when we get these testicles, it can be, we don't know how long the animal's been dead sometimes, and also they may be too young or too old, or there may be a disease issue. So sometimes the semen can be not viable anyway, but for this one, it worked well, to be honest. We've got the other things we can do is the oocyte collection. So we can actually take the oocytes from certain species and freeze them down. This can be quite difficult to do. We actually did this on a rare breed Cleveland Bay, and we managed to get people that did it managed to extract 8 oocytes, and they managed to mature 3 embryos. And this is actually a time-lapse camera, one of the first times it's ever been used out of the away from the human side, and it takes a picture every 20 minutes, and you can actually see these cells dividing for the first ever time. And they use this now routinely on the human IVF side to make sure these cells are dividing in the right order and the right consistency. So, but this is and then obviously they can take this embryo and either freeze it down or reimplant it. The other work we can do with the testicular side is if we can't extract the semen, or we can still extract semen, we actually can take a tissue sample from the testicles. Now, if you've got to imagine, wherever you take the samples from, those cells are programmed to do that job. So if you've got cells from the liver, obviously those cells are going to regenerate and produce the liver, and same with the kidneys and the skin cells, and same with testicles. If you've got a testicular sample, in theory, they're programmed to produce semen. So this can actually be used in a petri dish to produce semen, and this has already been carried out in mice and shown to actually be worked. Has it been done on many other species? Not that many, but it is a work in progress. But if we don't have those living cells in the first place, we can never do this. So this is a way that we can freeze the testicular tissue down to hopefully produce sperm cells in the future. And the testicular tissue contains obviously semifurous, gosh, I can't pronounce that now, the tubules, which can be cryopreserved, and the potential to culture the sperm cells, obviously from the testicles as well. The ovarian tissue, actually, it does exactly the same way. We can actually, the Rhino Fertility Project with Professor Susanna Williams has done this. So in the ovaries is all the follicles, and in the follicles, obviously, we can mature the eggs. So we slice these ovaries down very delicately and freeze them down. And this is a way that we can actually mature these follicles in a petri dish to produce the eggs. And this way, then you've got the eggs, they can be frozen down, or they can be used for ICSI or oocyte transfer as well. So this is just another way that we can use these reproductive cells. And this is the work that, say here, Professor Susanna Williams has been working on at Oxford University, called the Rhino Fertility Project, just to see how she can actually do this work for these amazing species out there. So, and we've had various different species in the last year. A tamarin monkey, a gibbon, and a cheetah have all come to us, females that have sadly passed away, but we can not only get the skin samples, we can actually get the ovaries and slice the ovaries up and freeze them down, and hopefully in the future be able to thaw these out to be used to be able to do egg collection from these in many years to come. Using skin cells, we can use these to be able to culture and then cryopreserve. As I say, this is one of the easiest cells to preserve down. We've got up to 5 days, the beauty about this. So if an animal dies, you know, yes, the sooner we get it, the better. But it's literally, you can even take, you know, 3 6-mil biopsies and send them to us as skin samples, and that's even good enough itself. But usually part of the ear is the best place to freeze it down to reuse it. So this is where the science really gets going, and we can either use this in a few different ways. We can either use these cells for the cloning purposes to get the DNA, because in that cell is your unique DNA. Or, as we were talking about before, these cells are programmed to do a function. But if we can reprogram those skin cells to induce pluripotent stem cells, and then by adding different growth factors, you could actually change those cells to grow into a sperm and an egg. So it's amazing what this technology could potentially hold out there. And already this work's been done in mice. So I really think the potential out there, again, is fantastic for literally storing down a skin cell and what we can do with it. But if we, again, I keep reiterating, if we don't have those living tissue cells in the first place, we cannot do this work in the future. So these skin cells can be transformed to pluripotent stem cells, and we can actually do this here by, we can actually grow the cell lines here from these skin cells, just to prove that those skin samples are able to regenerate, and we're already doing this with certain species already. So this actually proves that the sample is viable and can be used in the future. As I said before, it can be used for the cloning side. So in each skin sample is your unique DNA, and how this works on the cloning side is if we, from a donor animal, if we can extract the egg, take the nucleus out of the egg and replace it with the nucleus from the skin sample, and by an electrical impulse stimulate it to grow by an electrical fusion, and then this is programmed, obviously, the cell to, or this egg, to start dividing and growing. This is again being done in the domestic side with the cats, the dogs, and the horses, and as you see later on, actually it's starting to do this work in other species. So this can be used here and now to help certain species that have got you know, on the brink of extinction or need a new genetic line bringing in. Sex and semen, this is another application that we've used here. So in one jacket, you have the X and Y bearing sperm cells. And in the equine world, it's about 4% more DNA in the X-bearing sperm, which is the female sperm, than the Y-bearing sperm. And we can home in on that trait they've got the sperm cells are passed through a magnifying glass. It's about 20,000 sperm a second going through here, which is quite a rate that they go through. And they add a fluorescent dye. So with a fluorescent dye, the X-bearing sperm are going to shine about 4% brighter, and they're sent an electrical charge, be it positive or negative charge. And as they come through this flow cytometer, you have two magnets, a positive and negative magnets, and obviously opposites attract. So the negative ones with a negative charge will go next to the positive magnet and vice versa. So this is a very efficient way. It says 90% accuracy. We're normally up to about 95% actually. And instead of inseminating about 300 million sperm cells, we're actually doing only 40, but it can work very, very well. This next video shows us one of the first, well, this is the first ever foal born by this for a rare breed, a Suffolk Punch. So there's only 72 females left in the country. This just shows you a bit about it. And after 11 months of waiting, Ruby has produced a filly foal. And it's not just any old foal. This is a foal born by sex semen. We believe it's the first ever in the world for a rare breed animal to use sex semen. And using these technologies can really help this breed. There's only about 70 or 80 females left in the country. There's only 300 left in the world. They need all the tools of the industry, and just quite incredible now what we can do with science and technologies. And using this technology, maybe, just maybe, we can help stop these breeds from going extinct. But this holds the future. This little girl here. So there's the first steps. This little girl. And as you can see, it's all about partnerships, and just to get that one foal on the ground, it took an awful lot of effort from an awful lot of different people to be able to do it. And why we're talking about this side of it is purely because obviously these technologies from the rare breed side can now be used on for rare and endangered species. And Nature Safe, again, it's very much about partnerships, and we need your partnership really to make it work. So this is the Rare Breed Survival Trust Chief Executive said that this project demonstrates the viability of using new technologies to select female foals to increase the breeding population more rapidly than would normally be achieved in traditional methods. And we hope that will prove a model for more projects in the future. So the next sort of projects we're doing with this is to actually freeze it down rather than doing fresh conventional insemination, to actually freeze this down as sex semen, so we can use it for the future, male or female, on this way. So I think proof of concept really with, Kirk was born on the 10th of August last year, so he's just over a year old actually now. This is a Brzezowski horse. They are very inbred, and this was born through the cloning method and was taken from cells taken from 40 years ago at San Diego Zoo. So obviously this improves the genetic diversity of this particular species and really demonstrates that the losses of endangered species can be reversed by modern reproductive technology. So this is really exciting when this foal was born. And it's not all about, I know I've talked quite a bit about equids on this presentation, but it's obviously not all about them. We have the gorgeous Elizabeth Ann was born on the 10th of December last year, a black-footed ferret. And again, they were obviously nearly wiped out in the 1970s, incredibly inbred. And this was taken from cells 30 years ago. And I always think back 30 years ago, you know, could they ever possibly think, oh, all these technologies being used, when would it can be used? And it's great to be shown that it can be used and will be used in the future to help reverse the cycle of some of these rare endangered species, really. And it's a way that it can be used. So what have we done so far with Nature Safe? We've done about 116 different animals from 59 different species. Our target was 75 in the year, where we're about sort of 9 months in, so we should easily hit that target. And it's not all about just some of the more well-known ones. The mountain chicken frog, we've done a couple of those, and so on. The cheetah, and you know, some turtles, and obviously a Komodo dragon as well. So we've got a great list of different species we've done right across the board, and it's just how you can help. We sort of really reaching out to you watching this today. We'd love for you to be involved in any way you can. Primarily, obviously, we want the samples, so we are reaching out to the zoos and the zoo community to really get them on board, and we'd love to hear how we can collaborate with yourselves. So, you know, if you can spread the word and be a part of Nature Safe, we'd love to hear from you, really, really would. We can work out a memorandum of understanding together, an MOU. People quite often ask us, what's going to happen to those samples? Those are samples that are sent in are owned by the zoos themselves, owned by yourselves. So we can't do anything with those samples without your permission. You know, EASA can see what's in the tanks. We're very much obviously EASA accredited, so hopefully you know that we're obviously following all the right protocols and how we're processing, storing these samples as well. And we provide all the submission forms, we provide all the packaging, and it's very straightforward for you to do. And as we reiterated earlier, this is a free service. The only things we ask you to do, whether you can just post these packages or these samples back to you during the week. So it's fairly straightforward. When an animal passes away, you just clean that area. We can send you a full pack out and exactly what to do. Following the death, we have these boxes we can send you out 10 at a time. It's not a problem, with some ice packs in, and literally just post them back to us with a submission form. So again, when it comes to the epididymal side, it's a little bit more complicated, but the skin samples, very, very easy to do. So if you are interested in coming on board with Nature Safe, I say we'd love to hear from you after this presentation. And yeah, our vision is really to roll this out, our cryopreservation technologies globally. You know, we very much, when we got the knowledge, we wanted to disseminate it. This work we're doing, you know, is fairly straightforward under the right conditions with the right mediums and everything else. And so we need everybody's help out there all over the world to make sure that we can cryopreserve as many of our rare endangered species from all over the world. There's so many to do to bank. We would like to be the equivalent of the Millennium Seed Bank, for instance, so we can store these samples down from as many different species, so it's there for future generations. So really, thank you for listening. I thank you, Rhiannon, for putting this presentation on and letting and building the presentation so I can put it over to yourselves, and really all the parts of Nature Safe team. But really, more than that, thank you for listening, because we really would like to hear from you and be a part of Nature Safe and Nature Safe future. So looking forward to being in touch.